Photosynthesis. Now, photosynthesis is the process in which plants use sunlight to make glucose. Chemically, the equation is 6 water molecules plus 6 carbon dioxide molecules plus sunlight yields 1 molecule of glucose and 6 molecules of oxygen. Now, we know that the water comes from the roots and the carbon dioxide from the underside of the leaf and the sunlight from the sun, but how does the plant take that and turn it all into glucose? In the plant, all the action happens in the leaves. Now in the leaf there are many different layers, but the only one you need to worry about is the mesophyll layer. Now as you may recall from the anatomy of a plant cell, there is one special organelle where the magic happens, called the chloroplasts, and the mesophyll layers are the only cells that have the chloroplasts. But is this really magic? So in the chloroplasts, there are stacks of discs called the grana, and the individual discs are called the thylakoids. The liquid space that surrounds the cell is called the stroma, not to be confused with the stoma, the small openings in the bottom part of the leaf in which gas can be exchanged. Now back to the chloroplasts. The first part of photosynthesis occurs in the thylakoid membrane. This is also known as the light-dependent reaction. This membrane has a pigment called chlorophyll, which allows the plant to absorb photons in sunlight, as well as give the plant its green color. In the membrane there are two photosystems, photosystem 1 and photosystem 2. However, photosystem 2 comes first in the process. So, photosystem 2 takes the water brought by the roots in the stem and breaks it apart into oxygen, hydrogen ions, and electrons in a process known as photolysis. The oxygen atom combines with other oxygen atoms to form an oxygen molecule. The excited electrons go through the electron transport chain and go through the proton pumps where they can use the energy to transport the hydrogen ions to the other side of the membrane. This later creates an electric gradient to then use to move the hydrogen ions through the ATP synthase, which uses that energy to charge the ADP into ATP, which is usable energy. The electron keeps moving along the electron transport chain and gets excited by photosystem 1. After all that excitement, it gets carried by the electron carrier NADP+ to create NADPH. This NADPH then moves to the second part of the photosynthesis, the Calvin cycle, or light-independent reactions. This is where carbon dioxide comes in. In the stroma of the chloroplasts, the carbon dioxide molecules combine with a 5-carbon molecule called ribulose 1,5-biphosphate, or RuBP for short, using the Rubisco enzyme, to create a very unstable 6-carbon molecule with a ridiculously long name, which immediately turns into 2-phosphoglycerates, or 3-PGA. One of these flies off out of the chloroplast while the other stays inside the cycle. This PGA molecule is added to more carbon dioxide to make a 4-carbon molecule using the ATP and NADPH, and then more carbon dioxide to make the 5-carbon RuBP using more NADPH and more ATP. Remember that lonely PGA molecule that exited the cycle? The Calvin cycle just doesn't go once, it occurs over and over again, so this little PGA molecule has friends! No, not those friends. Yeah. So the PGA molecule gets excited to the PGAL state by ATP, and then combined using an enzyme to form glucose.
Are you getting this, Shaker? Are you getting this?
Yeah.
Alright, now what do we do with the glucose?
I know.
Okay.
Sit down. Here's how it works.
Cellular respiration. The reactants of cellular respiration are glucose and oxygen. Here's the equation for cellular respiration. C6H12O6 plus 6O2 yields 6CO2 plus 6H2O plus energy. The purpose of cellular respiration is to convert glucose into a usable form of energy. This form of energy is known as ATP. The first process of cellular respiration is glycolysis. The purpose of glycolysis is to split glucose into a smaller form so that it can enter the mitochondria. This process takes place in the cytoplasm. Glycolysis results in a net gain of 2 ATP, NADH, and 2 pyruvic acid molecules. Pyruvic acid enters the mitochondria and loses the CO2. It is then combined with coenzyme A to become acetyl-CoA. Acetyl-CoA then enters the Krebs cycle, where it will eventually break down into CO2, releasing energy and electrons. These energy and electrons make ATP, NADH, and FADH2. Playing the role of electron carriers, NADH and FADH2 carry electrons to the electron transport chain, which is located in the membrane of the mitochondria. The electrons are released by NADH and FADH2, which become NAD plus and FAD again. And when these electrons go down the electron transport chain, they release lots of energy, which converts ADP to ATP. The electron acceptor at the end of the ETC is oxygen. Oxygen combines with hydrogen to form water. But when there is no oxygen available, cells can go through the process of fermentation. Fermentation acts like a backup process. The purpose of fermentation is to convert pyruvic acid to lactic acid for muscle cells. This is known as lactic acid fermentation. However, in yeast and bacteria, cells convert pyruvic acid to alcohol and CO2, which is known as alcoholic fermentation. This is used to recycle NADH back to NAD+.